I really enjoy working with Dr. Strauss. I really enjoy being able to really dig in and actually design my own experiments and really think like a scientist. So this research has been very helpful for me deciding what I want to do in the future. So first we have to remove our U and cut the substrate. This is going down over time. And then we have to incorporate our cytosine. Now this is adults. This first one right here is adults. We knew this was going to work. This proves that our assay works because this is well characterized in adult tissues and cells. So we know basic excision repair works in adults. My future goals are actually to go to medical school. I've been interested in that in a few years now, but I believe research is something that doctors need to understand and be conscious of, because every drug and treatment that is developed comes from the bench, and then you have to bring it to the bedside. So it is very important to understand that link. And so when I eventually practice medicine, I'm glad to know where this stuff comes from. So, very good.
Experience has helped me realize what actually goes into research. I think research is actually a very big possibility for me. I'm still debating whether it would actually be better for me to do that than medical school, but I really do love what I've done, and I do think that I would love a career in this at some point. This is how we measured the metabolic rate, which would be the energetics of the running and walking. This is a human, basically the measurement of the oxygen consumption through this little mask, and then it goes into the oxygen analyzer, and we can find the metabolic rate through this. Here, as the kinetic is decreasing, that's where the foot's acting as a decelerator. The body is, again, the potential energy is increasing. So these two are out of phase, as was explained in the last slide. So for our studies, we're trying to relate this mechanism of walking to the amount of energy input for this walking. We studied Alba lactuca, which you can see here. It's a green algae or sea lettuce, and it's found in the intertidal and subtidal in New England. And we wanted to look at the uptake of polychlorinated biphenyls and the possible use of alba as a remediation agent. And here's New Bedford Harbor itself. It's actually a very well-known fishing port, and this is where it comes in. Where I'm from, I actually grew up in Fairhaven, which is right here. So my father's a tugboat captain, and I grew up playing in this water and going out on boats and just sort of not thinking it was too big of a deal, because we weren't taught that it was a big deal until now, when I find out all this information and how toxic this water actually is. Basically, we collected seaweed from both Nahant, Massachusetts, which is a clean site, and from New Bedford, the polluted site. We cleaned these cultures, made sure there was no bacteria or any other organisms on it, and basically we stuck it in these vials and spiked in a certain amount of PCB and let the alba uptake the PCB into its system. And once we let this happen for a certain period of time, we then analyzed the tissue and the water by GCMS to determine where the chemical was, whether it was in the water or in the seaweed, and to what extent it was in there.
I plan to go to graduate school for bioinformatics, and this is extremely important because chemical informatics is new and evolving, and any sort of protein software, I can tell now using the software that it's not very good at predicting any sort of structure. So maybe with the things I learned at the Engen lab, I'll be able to help create better software. So this is a diagram explaining the HIV infection. Here you see the HIV viron, and it becomes encapsulated into the normal, this is a normal cell. It releases a capsid, which you see here, which releases RNA because it's a retrovirus. The RNA becomes DNA. So what about HIV-2 VIF? Its mode of action is very poorly understood, and there's much, much less research than there is on HIV-1 VIF. There's no conformational data at all, and it's also analogous to VIF and HIV-1, and it can even functionally replace it.
I'm graduating in May, and I'm planning to work in the biotech industry for a year or two, and before I go back to graduate school. I really want to find a topic that I'm passionate about and to commit 5 years of my life.
Today my talk
is on the dynamics of the polymerase manager protein UUD, and we're going to look at DNA damage tolerance in E. coli. The Y family polymerases are able to bypass lesions, and they lack 3' to 5' exonuclease activity, and they're also inherently less they're prone to mutations, and so they must be tightly regulated and only be working under conditions of DNA damage.
I have a job lined up at Children's Hospital right now to work in a neuroregeneration lab. Neuroscience is another passion of mine. And ultimately, I would plan to apply this following cycle for graduate programs in computational neuroscience. And so the general problem is that structural genomics projects are expressing proteins from the genome on a high throughput basis and finding their 3D structures. So there are about more than 3,000 structures that are annotated as hypothetical and with unknown functions. We don't really know what they do, even though we have their 3D structures. So what we're interested in doing is finding ways to predict the function of these structures. Thematics has a fairly high success rate, and it has superior precision, especially to other 3D structure prediction, functional site prediction techniques with a very low false positive rate of about 3%.